undergraduate programs, but I also initiated and teach some of our high school programs that are run in the summer. I'll talk a bit more about that later, and assist with some program development and projects as well. So this background picture is actually from the hill over there, looking down at our school. This is one of our new academic buildings. This is our academic center. There's a number of classrooms and labs and spaces that we use for everything from the graduate classroom lectures to interactive spaces to events like this one. We also have a residence hall immediately across the road right there. That's also brand new, and a dining hall. So it's a good place to be. It's a little tiny residential campus located in the middle of the Smithsonian Conservation Biology Institute, which is a 3,200-acre research facility run by the Smithsonian and with the National Zoo, where research on endangered species takes place, and a lot of forest ecology and other types of conservation research take place. So what happens is we get students from all over the country to enroll in our courses across different age groups. Starting with 5th through 10th grade with the Fawns Nature Camp that is housed in the residence hall over the course of the summer. And we have courses for college credit for 10th through 12th graders that take place here in the summer as well. And those have not been finalized for this year yet, but keep an eye on our website and talk about that later. For the undergraduate programs, what we do is kind of like a semester abroad. Students come and live here for 4 months for a full semester and take all of their courses here on site. And because they're residential students, we can engage them all day long. We're not limited in terms of a typical college schedule where they're here in class from 9 to 10 on Monday, Wednesday, Friday, which allows us to spend a lot of time getting really in-depth in different types of conservation case studies and scenarios. So it's a pretty awesome deal for them. We also offer courses for graduate students and professionals in the field. A lot of those are techniques-based, and some of them are project management-based. And those typically run 1 to 2 weeks long, and the students, again, live on site and engage with professionals and network. It's a pretty good deal. So that's a bit about who we are. In terms of my background and how I got here, I grew up in upstate New York. I grew up there. It looks like that. There's tons and tons of little lakes, which is something that's strange to me about Virginia, that there aren't very many lakes here, but where I grew up, there were a lot of them, and I spent my time playing outside and really exploring, and that was the central part of my life. I like to explore. I like to see new places, investigate. I like to imagine that the world had been not yet discovered, and I was the one who was finding all of the animals for the first time, seeing all the plants and areas for the first time. And so that was sort of a central part of my personality and upbringing. I was lucky to go to school also in upstate New York at Cornell University, where I had the opportunity to engage in a lot of different types of research projects. And at that point in my life, I thought that I was going to be a field biologist. That was my life's goal, right? I wanted to be a field biologist and explore new animals and species and habitats and really get a chance to see what the world looked like. And I had the opportunity to work on a couple of different projects, or a few different projects that were really inspiring to me. The first that I worked on had to do with, this is an elk. We were doing plant surveys in areas where there were reclaimed strip mines, so areas where coal had been removed and then they had gone through and tried to restore the environment that was present in that area by seeding it and putting in grasses and early successional plants. And what we were really looking at was a reintroduction program with elk and seeing where the elk were choosing to have their young and whether it would be successful for that species to end up being a sustainable population. The next project that I worked on had to do with behavioral ecology. I was really interested in birds. I thought I was really interested in behavior. I ended up working on a really cool project that looked at song types in banded wrens. They're closely related to Carolina wrens. That's why they look a little bit familiar. But this is a project, this is a species that takes place in Costa Rica. So I got to travel to Costa Rica and look at the way that they use different types of song to communicate information to each other about their territories and their mate selections and things like that. It was a really good time. But I learned from that I'm not that interested in behavioral ecology. So I ended up working and doing my honors thesis as an undergraduate on stream ecology in New York temperate streams and looking at the role that different types of invertebrates play in nutrient cycling in streams. So that was really an important project for me, not because I am an expert in nutrient cycling. I've pretty much forgotten most of what I learned in that, except for the basics. But because it made me attractive to graduate schools. And I knew that, again, I knew I wanted to do research. I knew I wanted to be a field biologist. I didn't know exactly what that would look like for me. But I started applying to graduate school. And in the end, what ended up happening is I was accepted to a lab that was working on deep sea issues. And this was perfect because, again, that passion for exploration and discovery, we have explored only a tiny fraction of our oceans. And that's not to say that we haven't mapped our oceans. We've mapped them to various levels of resolution. But only 3 to 5% of our oceans have actually been seen by human eyes, right? Now, we know the coastal systems pretty well, but we know almost nothing about the deep sea. We know very little about the deep sea. And so this was a chance to get to know that area, and I was so excited to do this. So I started as a master's student and switched to a PhD program on projects that looked at cold seeps and corals in the Gulf of Mexico. Everybody thinks they know a lot about corals, the shallow water corals that are so beautiful and colorful and really important for fish and other species. There's actually deep water or cold water corals as well that live far below the reach of sunlight, and those animals are also extremely important as habitats for fishery species and other types of organisms. So we were looking at the corals and at the sites in which they're found, including cold seeps. Cold seeps are areas where oil and gas seep out of the seafloor, just chemicals seep out of the seafloor, and actually end up forming the basis of a lot of really interesting chemical reactions that help to support life through chemosynthesis instead of photosynthesis. And what this map shows you is the Gulf of Mexico. Louisiana's up there. And this is the continental shelf in the Gulf of Mexico. And the yellow dots that you see here are places that the lab had surveyed before. They are places that had been visited before, even if only once or twice. They've been photographed, they've been seen. The areas that were red were areas that were predicted to also have life, but nobody had ever visited them before. So I was coming into the lab at a time when we were going to be exploring these sites for the first time. It was a really exciting time to visit. Plus, cool animals. Just got to show some pictures, right? So these are the kinds of organisms that we're seeing down there. You can see in this picture, you can see there's some tube worms, there's some octocorals, there's some brittle stars clinging to them, a little crab in the middle there. I love this animal. It's one of the most beautiful animals I've ever seen. It's an octocoral again. It's not a reef-building coral, but it's another type of coral. You can see little shrimps swimming around it. If you've ever flipped over a rock or a log and you've seen roly-polies or pill dogs or icepods, this is a relative of that animal, except they get to be about a foot long. There's a couple different strategies towards survival in the deep sea. You can become very big or you can become very small. There's not a lot of food down there, so if you become very big, you have the ability to migrate to different areas to find food. If you become very small, you have fewer resource needs. This guy got big. They're ubiquitous throughout the world's oceans. They live in soft sediment, they dig little burrows, they have extremely sensitive, basically a smell, right? They can pick up traces of the chemicals of water and then move to that area and eat the fallen whale or fish or whatever it is that's fallen to the seafloor. They're really cool animals. In order to explore the deep, this is beyond scuba diving depth. So we used submersibles, we used the Alvin, we used the ROV Jason, we also used one that's not pictured called the Johnson Sea Link. And basically the machinery allows you to get a really good understanding of the site since you can't go out and walk around as much as I would like to. The manned submersibles like the Alvin allow you to really get a good look at the site yourself. But actually the ROVs like Jason give you better data. They're more stable, they're easier to control. And that's the inside of the Alvin. If you can imagine my height, right? So I'm standing at the base of a metal sphere. The top of my head is touching the top of the sphere. So if you spin me around my middle, that's the entire space. And there's 3 people in there, along with all of the instrumentation and equipment. It's very crowded. And basically you go down for about 8 hours. It takes about 2 hours to reach the seafloor. You spend about 4 hours or so on the seafloor, 4 to 5 hours, then it takes about an hour to come back up again. So it's an experience. The remotely operated vehicles can stay down for 36 hours or longer, so you get a really much better look at the site. And you can do that by using computers and cameras that are mounted on the outside of the submersible. You're sitting basically in a trailer that's called a control van, and you're looking at these monitors and getting a sense of what's going on in the environment around you. So you can see front, back, sides, there's video, there's cameras, there's different types of chemical sensors, there's little sampling tubes and suction devices so you can sample animals. It's pretty neat. There's a sense of wonder in the deep sea that's hard to describe until you've been there. And I like this picture because it tries to capture some of that emotion. When you first descend in the water, the water's beautiful. It's like bright blue, right? The sunlight's shining through the water. Sometimes you see fish or whatever. And as you descend, it gets darker and darker and darker until you reach an area where there's no light, and that's when you start to see the bioluminescence. And people don't really appreciate how much bioluminescence there is in the oceans. So many different types of organisms produce light. Single-celled creatures, jellyfish, different types of fishes, and they all have different colors and tempos and rhythms, and it's like the most incredible firefly show you've ever seen. And then eventually you reach the very deep sea, where there's all these really cool, crazy animals. This was an amazing project to be a part of. And it suited also my desire to do pure science, right? Pure ecology, where I could really understand the undisturbed environment, look at the interactions between different types of organisms, and try to get a good sense of what's going on in the world. So that's how I spent the next 5 years. I looked at questions, basic descriptive ecology, general ecology questions. What's the successional patterns of these sites? Who's there? What types of relationships exist between the different types of species that are found at these really bizarre communities? And typically what you end up seeing is in an area where there's active seepage or a new seep. Seeps last for thousands of years. They're not ephemeral habitats like hydrothermal vents or undersea volcanoes, which only last for a few years at most. These areas can last for thousands of years. But if you see an area where there's a lot of seepage or where it's a new site, you see a lot of bacterial mats, like in this corner here. The red dots are lasers that we use for scale. I don't know how far apart those ones are, but usually they're 10 or 30 centimeters apart. Typically, after the seep has been established for a long time, there's areas of greater or lesser seepage, different types of chemicals present. You see a lot of mussels, different types of mussels, small mussels, big mussels. Some of the types of mussels that you see are very large, and it's always like, sure, you're tired. Look, it's my mussel. Yeah. And so those, you can find massive aggregations of different types of mussels that have symbiotic bacteria in their tissues that allow them to use the chemicals present in the seawater and in the sediments for energy. In other types of areas, or in older seeps, you find tube worms. And tube worms are these really interesting animals right here. They have no mouth, gut, or anus, right? So what they have instead is a tropisome full of symbiotic bacteria, and they have a really interesting symbiotic relationship with the bacteria in their gut and in the sediment around them, where there's anaerobic oxidation of methane coupled to sulfate reduction, which yields sulfide, right? And then there's sulfide oxidizing bacteria, and it's this crazy cycle. And they basically can have sulfide oxidizing bacteria in their gut that can break apart different chemicals, provide sugar to the host organism, the tube worm itself, and then they can excrete the byproducts into the sediment, the bacteria in the sediment, then use that to produce more more resources for the tube worm itself. So it's a self-sustaining relationship that can last for hundreds of thousands of years. And individual organisms can live for up to 600 years. They grow very slowly, they have very low mortality, it's a very low stress environment, very low levels of disturbance, they're crazy. I don't like collecting these guys, right? They're very old. So if you're thinking of like the ethics of collecting organisms, that's an old animal. You can see that there's also lots of associated animals in this area too. These are a bit like the bushes or the trees of the deep sea world. There's shrimps, there's crabs, there's all sorts of tiny little invertebrates that live on these things, hydroids. There's some clams in the background of this picture. It's a fascinating little community. They're like little tiny islands in the deep sea. Also present in the environment are soft corals or octocorals like this one that's not a reef-building coral, but you can see there's associated fauna on this as well. We looked at those as well. And we also looked at reef-building corals like Lophilia pertusa, which can form massive reefs in the bottom of the ocean. And these things are, again, important nurseries for fish. They attract lots of different types of organisms near them. They affect the way that the current flows. They're really interesting and important animals as well. And, you know, this was a great experience. It was fascinating work. And it made a big impression on me. We had the opportunity to see some of the most amazing animals I have ever seen or even heard of. Like this octopus who got really curious about the Jason manipulator arm. But this is what I also found at these sites. And these are sites that no human has ever been to, ever. We were the first people to go to that site, and we found trash in the bottom of the ocean, right? So this is a bunch of soda cans or beer cans, different types of cans, so probably, I don't know, maybe someone dumped over like a whole bag, I don't know, but they ended up at one of my sites, and you can see this is an urchin here that's burrowing through the sediments. You can see different types of mussels in the ground. So it's a you know, this is a community type where there's trash, and that surprised me. And then at the end of my degree, the BP oil spill happened. And in fact, some of the corals that we studied ended up being some of the corals that were affected by the BP oil spill. So that, again, really made a huge impression on me. Remember that I wanted to go into this field because I thought I was going into the pure ecology. I just wanted to look at an undisturbed environment. What's going on in this system? And that really sort of changed my mind. Around this time, I also started teaching. I started teaching, typical grad school courses I was teaching in the labs. There were a couple other classes that I was doing, and it was really fun and really rewarding, and I liked it a lot more than I thought I would. What's a success? Is a success here when you've been monitoring the population effectively enough to determine that it is declining, so you can maybe take some steps to save it? Is it here when the rate of population decline starts to slow? Is it here when the rate of population growth becomes higher, becomes more increased? Or is it there when it starts to approach the level that it used to be at? And honestly, this is unlikely. In any case where there's a species that's going extinct, this is unlikely because maybe the habitat is gone. I mean, there's a reason that it declined in the first place. So trying to figure out what your end result goal should be is pretty important too. What counts as success in this case? What we do in our programs here is we try to teach this approach to addressing this question, and it's a project management approach. And the old method, the old way of doing this was basically to say, hey, there's a population going extinct. I have a tool in my toolkit that can help with that. I have a method. I'm going to do my method, right? And I'm going to do it in this way. And if I have enough time and money and resources, I'll check and see whether it worked, right? This is a very linear model, and it's not going to be an effective strategy for figuring out if we have how to best save a species. A better way is a more iterative model, right? A more circular model where we can make a plan, put the plan into action, monitor and evaluate whether it worked, make changes to the plan, but that plan is, something more cyclical. And we teach a program called adaptive management, which is based on this project cycle management. We use as a foundation the Conservation Measures Partnership Open Standards, which you can find online, and I'll include a link to it in the documents. I actually have a handout myself. And basically what this is a cyclical model where you begin by drawing a picture of what the problem is. You try to understand the components of the problem. And the second step is you're planning your actions and you're monitoring, right? So those two things happen at the same time. The third one is actually doing the actions and the monitoring. The monitoring is not a tacked-on thing at the end, it's part of the process. And then the fourth one is you're analyzing, using, adapting, you're checking out whether it works. And the last one is communicating the results so we don't make the same mistakes over and over and over again. And the students that what we focus on with the students are really steps 1 and 2, and this is what I'm going to talk about today. We're trying to get them to identify what the target is, what their goal is. What are you specifically trying to conserve? We're trying to get them to identify what the main threats, the direct threats to those targets are. And then we're trying to get them to determine what the indirect threats that affect the direct threats are. Threats are always anthropogenic in this model because we can't control weather, right? There's things that we can't control. But we can, again, conservation is about people, we can control people, or we can affect people's decision making. Are these things always science-based? Usually not, right? So to get a good idea of what the threats to a species are, we have to understand the economics, the policy, the social systems that underlie the choices that people are making. So we put this into a picture. You can basically, again, this is exactly what I just said a second ago. You can identify the scope, the geographic area where you're trying to make decisions. You can identify your targets and your goals. And then you can figure out what the direct threats to all of those things are. And sometimes these threats are related. So this threat, for instance, affects this target and this target. Both of these threats affect this target. So you have to figure out sort of like how this thing is webbed together. And if you're doing this in real life, it looks pretty complicated pretty quickly. And then, of course, there's the factors that affect those direct threats. And I keep saying indirect threats. I should be saying factors. These can be positive or negative. These can be things that negatively affect the population, or they can be things that are opportunities for the population. Following this understanding or this conceptual model of the systems, the students move on to step 2, which is identifying strategies that they can use to reduce the direct threats and conserve their targets. So you have a target, you want it to be improved, you have a strategy, and that'll improve your target, right? There's some implicit assumptions that your strategy is going to work. So the biggest thing to realizing this model is that each step focuses on the achievement of results and has assumptions that can be tested. So when we put this together, we have, at the end, we have a goal, we have the impact and the target is our goal, we have a strategy that we think is going to affect that target, and we have intermediate results. So these are always phrased as results. So for example, we have a strategy, we think this is going to happen when that strategy takes place. And if it doesn't, then we need to revisit our assumptions and change our strategy, and this part immediately becomes its own little cycle. And this allows us to test all of our assumptions and make changes while we are engaged in the first round of action, right? So you can start this process with an imperfect understanding of your system, which is really important in conservation scenarios where you don't always have all the answers, and you can't just wait to say, oh, I need to completely understand the behavioral ecology of the species before I act. It's usually too late by that point. So here's a specific example. Let's say that we're trying, I'm sorry, this one is quite small. We're trying to conserve a forest in which hazelnuts grow, right? That's our goal. We want to conserve this, protect this forest. Our strategy over here is that we've got this management plan. We've got this idea that we have this plan, it's going to work, it's going to save this forest. And the first result is that the plan will be implemented. Okay, so that's a good first result, right? If you can't implement the plan, then it's right back to step 1. That will yield a higher quantity of grizzled nuts. There can be more nuts. It's going to be good. That will yield higher income for the people who live in that area, right? Which will give them a higher quality of life. These are all results. And if any one of these turns out not to be true, we can revisit the assumptions that underlie that. For example, if their income doesn't become higher, why is that? What can we do to address that in the moment? The higher quality of life will hopefully yield a greater recognition of a forest's economic value, which will hopefully yield local people committed to forest management, which will yield less conversion of the forest to agriculture, which is addressing the direct threat to this forest, which is that it's being burned down for agriculture, which will positively affect our goal. It will conserve the forest. And we have the students map this out for different types of scenarios. And what it does is it gives them a really good understanding of the different types of things that can influence a conservation problem. The science, the policy, the economics, the social systems, etc. You may notice it's quite similar to another method that I'm sure you're all familiar with, right? So it has a lot of similarities to the scientific method, which of course is a good thing. The reason that we use this adaptive management model is because it allows us to pull in threads from different disciplines, and it allows us to act with a perfect coalition. Let me give you a quick story where you guys have a chance to try this on your own. So BlackFooted Ferrets are a species that the SCBI has worked with for many years. BlackFooted Ferret, besides being really cute, they're one of the big success stories in conservation. Is it a perfect success? We can get to that. But these guys are a native ferret to North America. We don't have any other native ferrets. The ferret that you see in the pet store is not native to North America. They had a huge historic range. They were found in prairies throughout the lower part of Canada, northern Mexico, and all throughout the prairies of the United States. And they declined rapidly because their food source, prairie dogs, declined rapidly. And basically what happened is prairie dogs live in burrows. The area in which they lived was being converted for different types of agriculture. So farmers didn't like them because the area around a prairie dog burrow is bare of vegetation, so it's not easy to farm near a prairie dog town. And ranchers didn't like them because they believed that cows and horses would break their limbs by stepping in the holes. So they were exterminated by people who owned land, but also by the government who had a big bounty for prairie dogs. And that's actually a panel of prairie dog birds. They were exterminated in the vast majority of their range. And once they were gone, they're a keystone species, lots of other organisms depend on them. BlackFooted Ferret was one of the ones that declined really quickly. So So there were a couple other things that happened at the same time. Bubonic plague spread to these guys, so they're very susceptible to certain things. They, so that plague and these guys becoming humans. These guys were wiped out. They were really, really wiped out. And even today, the vast majority of their habitat is gone. It's urban development, it's farmland, or it's ranches. We don't have the massive prairie ecosystems that we had previously. In the 70s, the black-footed ferret was declared extinct. They were no more, and people just assumed they were completely gone. They hadn't been seen in a long time. In 1981, a dog found a black-footed ferret, brought a black-footed ferret home to mom and dad, and actually it's a funny story. The dog's owner threw the animal out. I was like, oh, gross, a dead animal. The wife looked at it and was like, I've never seen that before, and took it to a taxidermist who identified it as a black-footed ferret, and sent the sample off, and a remnant population of a few dozen animals was found. It was carefully monitored. It was a very exciting discovery. It was carefully monitored and eventually went up to about 128 individuals before it crashed. The population declined very quickly. And in 1985 through 87, there was a capturing program to try to bring them all into captivity for a captive breeding program to try to save the species. So the ferrets were captured for captive breeding. I'm going to save you the details of the whole story for their captive breeding, but it was a massive effort on the part of dozens of scientists across the country, including here, to try to get these guys to have a sustainable population with, I think, like 17 founder individuals, very low genetic diversity in this population. But it worked. We got black-footed ferret babies, which are pretty freaking cute. I think that's adorable. So, they were successful enough in their captive breeding efforts that by 1991 they were able to reintroduce some individuals into the wild. That had limited success. So as, you know, again, adaptive approach where you're looking at what works and what doesn't work, they were able to ensure increased survival by vaccinating these guys. They're also, again, susceptible to diseases, including canine distemper, which did a number on the population previously. And with preconditioning to allow these animals to live in little pens where they have to, before they can be released, they have to show that they can kill a prairie dog. They're not going to make it if they can't kill a native prairie dog. But it gives them a chance to learn some of the behaviors that they'll need in order to survive in the wild before they're reintroduced. There's now 27 reintroduction sites throughout the prairies on private and public land, and some of them are even self-sustaining. There's enough individuals in that population that they can self-sustain, which is a big deal. Most of them are not self-sustaining. They're being replenished with animals that are bred in captivity, right? So now there's thousands of ferrets in population through captive breeding efforts and through the wild ones that are breeding. But it's by no means done. We're not finished. We're probably going to have to manage the species forever, right? That's a value-based decision. That's somebody who's saying, I'm going to expend resources on saving this species because we think it's important. That's an ethical decision, which makes it, again, sort of different than what's going on in the pure sciences, right? If you're looking at a pure science approach, they don't got it, they're not going to make it, right? We're deciding to expend resources on these guys. So that's sort of an interesting case study. What's the status now? We need more land, we need more habitat. We need to be able to work with landowners to say, okay, we want to protect not only the black-footed ferret, but also the prairie dogs. And these prairie dogs are, again, a reservoir of disease. Is it worth it to bait prairie dogs with little bits of food that have vaccines in them? There's trials going on now to see if we can vaccinate prairie dogs. I mean, isn't this sort of an interesting question? So, if we were to build an adaptive management model for ferrets, what would it look like? What's the goal? What's the target in this case? What's the geographic scope we're going to work in? Right? It has to be manageable. It has to be something that we can function in for a reasonable amount of time. What's the direct threat? What are the big #1 threats to the species? What are the indirect threats that influence the direct threats? What are the factors? What are the opportunities? What stakeholders need to be involved? Who needs to be sitting at the table to make the decisions about these guys, right? So a lot of this is government program. It's sort of interesting. What are the strategies that we need to address this program? What are the results that we expect based on these strategies? So, again, visually, you build a conceptual model. You have the targets identified, the geographic scope and your goals. You have the direct threats and you have the factors that influence those threats. Second step, you build your results chain. What is your goal? What is the strategy? What are the results that you expect, and what are the assumptions that underlie each of these results? So if you were going to do this, we have a couple minutes left, so I'm really happy actually about that. If you're going to do this, what should the goal be? Does anyone have any ideas? What's the end result? Yeah.
Self-sustaining population.
A self-sustaining one?
Well, good
One
point.
population?
Multiple self-sustaining populations.
Okay. Any other ideas? Do people agree? Yeah? Okay, so what's the geographic scope for that? Do the populations need to be connected? Because I don't actually know which of these 27 they are. I should have highlighted them, but I'm not sure which ones they are. I don't know if they're close together or not. I suspect probably not. Yeah.
I would say no, because the world is a different place now than it was when they were on their decline.
Exactly.
So I would think you'd want this population to be the Nevada population, this population would be the Minnesota population, and yeah, I mean, it'd be interesting to study that to see like how the populations change because
Sure.
they're adapting in that specific time.
Okay, good. So what are the direct threats to the population? Yes.
Well, if you had them together
Yeah.
and there were diseases, I would want to have them spread out.
Yeah.
Because
that's an
then you
excellent
wouldn't
point.
wipe out the whole population of them.
Yep, that's an
They'd
excellent
have
point.
separate ones.
Particularly in the self-sustaining populations where we may not catch them all to vaccinate them, right? Yep. So definitely there's protection in having several small as opposed to a single large reserve. Yep. That's one of the big controversies in conservation is how do you design reserves or parks? Is it better to have lots of small ones or single big ones? I mean, ideally you'd have several big ones, right? But it's often not an option.
Well, and there may be different diseases in different, that would affect different populations differently.
Definitely.
So your inoculations might even be, your vaccinations might even be different,
Absolutely.
depending on the population and location.
The two big ones that we know about right now are the plague and the canine distemper. Those are the two huge ones that affects everybody. But you're right, there could be others that we don't know about yet. Absolutely. These are great comments.
People, we don't want them on their land.
Yeah, big threat is people. There's still a huge amount of prairie dog hunting, right? It's been pretty well established at this point, just to clear up the misconception about broken limbs and holes. Horses and cattle don't step in the holes and they don't break their limbs if they do. In fact, having prairie dogs in a setting like that actually makes a healthier ecosystem. It's sort of one of those interesting fallacies.
But do the agriculturalists believe that and know that?
They don't really believe it, but
So
there's-
education programs,
Yeah, absolutely. That could be one of the factors. There could be a strategy that would yield an expected result of people maybe deciding not to shoot them.
right.
These are great. Yeah, that's exactly the kind of thing that we would be doing for the species. The thing that I want to remind people is that it's an iterative process. So for example, if you put together an education program, perhaps what it would do is actually teach people where the colonies are. Maybe suddenly there's a market for prairie dog pets and birds, but maybe there could be, right? And so we start seeing increased harvesting. That would challenge one of our assumptions that if people know more about the species, that they would be more inclined to protect it or not hunt it. We'd have to go back and revisit that strategy. So it's always got to be an iterative process. And in my last couple minutes, I just want to conclude by saying the science content is hugely important. There's no way we could protect prairie dogs if we didn't have an understanding of how to vaccinate them, how to breed them in captivity, of their reproductive biology, of their behavior, of their ecology. All of that is crucially important. But none of the conservation efforts would have worked had there not been an interdisciplinary approach to saving the species. And more important than even the science content was the effective communication that led to people having the space and protecting the habitat and saying, okay, on my property you can bring in black-footed ferrets and prairie dogs. The teamwork and the project planning. All of those things are so important. So I guess what I want to encourage people is to think about sort of these, they're called often soft skills or non-cognitive skills, which is an expression I hate. But these types of skills, especially for scientists, I think is so crucially important in order to enact conservation, where you have a big mission, right? If you're trying to save the world or save species, these types of skills are really crucially important. And a little plug, this picture is actually from my high school class from last summer. It's something over in your high school class, actually. The programs that we offer tend to be field-based, field ecology-based kind of events. One of our guests actually had a herpetology course last summer as well, based out here. So I encourage you to keep an eye on our website. And if you're interested, I also have a flyer with some information about the Smithsonian-Mason School of Conservation as well during the break. And with that, thank you very much for your attention, and I'm happy to take any questions.